Okay. So thank you for joining us for the Kirby Institute seminar series. My name is Chan Fetzifan. I'm a senior lecturer in the Immunovirology and Pathogenesis program at the Kirby Institute. Before we begin, I'd like to acknowledge the traditional custodians of the land in which we are gathered today and pay my respects to their elders past, present, and emerging. Today, we are presenting from the Bidjigal lands of the Bidjigal people of the Eora Nation, and I extend that respect to Aboriginal and Torres Strait Islander peoples joining us today here in the audience and online. So before we begin, there's some housekeeping to start off with. The format of this seminar is a presentation followed by a Q&A session afterwards. If you're joining us online today, you can ask questions by clicking on the Q&A icon at the bottom of the screen and then typing in your questions. And then for the audience in the room, we have a microphone at the side there for you to ask questions at the end of the seminar. So now I'd like to introduce our speaker for today, Dr. Jelke Fros, who's an assistant professor at the University of Wageningen in Netherlands. Jelke did his PhD at the University of Wageningen and then did his postdoctoral training at the University of Oxford, where we met at the Peter Medawar Institute for Pathogen Research. And there Jelke worked with Professor Peter Simmons on viral RNA, and then is now starting up his research group at the University of Wageningen, looking at dual-host viral life cycles with a particular emphasis on arboviruses. So I'll hand it over to Jelke.
Right. Thank you, Chan, and thank you all for joining us today. Let me see if I can share my presentation with you. There we go. And get myself a pointer, perhaps. That's not bad. Pointer, yeah. Okay, well, we can do it without, no worries. Yeah. All right. So before we start talking about any of this, I thought it's nice to maybe introduce myself a little bit further and why I'm here. So my name is Jelke Fros, and I am from the Netherlands. And we're actually here for 7 months, me and my family, because my wife got a nice fellowship at St. Vincent's Hospital. And thanks to COVID, I'm able to actually lead my research team for these months via Teams. So that's all great. And that also allows us to spend the weekends with these two little monkeys in sort of nicer surroundings than what you usually get in the Netherlands. So for those of you who are a little bit unfamiliar about this, so the Netherlands is this tiny little country in Europe, and just measuring across is about 80 kilometers. So it's actually really, really small. And you can see Wageningen, which is the city where my university is a little bit in the middle on the east there. And that's about 45 minutes away from Amsterdam. And this is what our campus looks like. So here you see what a lovely scenery the Netherlands has. There is not a single hill in sight. And we do have a little bit of grassland and a little bit of farms in between the cities, but it's very densely populated. So here we see our campus, which is actually a really nicely set up campus. And if you see that big square red building in the middle, just behind there's a building with little green windows there, and that's our building where we have our virology. Now, our university is usually, historically, is really good at agriculture. And behind there, you still see a lot of fields that belong to the university. So they do a lot of agricultural sciences. But nowadays, there's a lot of good biotech, life science, zoology, and a lot of research, climate change, and other sustainable sort of research. Right. So why did you heard Chan say, I did my PhD there, and then I moved back. And there's one main reason why I want to set up my group there, and that's because we have a lot of plant research. We have greenhouses, and we also have fantastic entomology groups. So a lot of research into insects. And together with the entomology group, we've previously set up this BSL-3 laboratory where we can do infections with live mosquitoes. And that's something, so that's the picture you see on the right. And that's how we dress usually there. And that's just so a mosquito, if we by accident let one escape, which has never happened yet, it cannot sting you where you can't see it. So that's the precaution that we take. And my team has recently been growing. So I now have 6 PhD students and a 7th is starting next month. And they all work sort of on how mosquito-borne viruses are able to transmit their viruses, what sort of pressures are on the viral RNA, either in the mosquito or in the vertebrate host, and a lot do things with biotechnology. So we look at protein expression systems, how we can improve that, how we can use this knowledge for vaccine development. And this girl on the right, she is the craziest of the bunch. She actually tries to get unnatural base pairs into a virus, so she can use those, so she extends the genetic code to have new base pairs in there as a new sort of biosafety mechanism. I will not talk any further about that because we just got these projects started, but last week she showed me her first virus with unnatural bases in it. So she is actually making some really good way, and hopefully soon you'll hear about this a little bit more. But for today, I really want to focus on the viruses and their vectors, and how these arthropod-borne viruses, how they can switch between very distinct host species. And the focus in that is mostly on how the viral RNA is recognized and activates immunity. And we've been dabbling in some other viruses as well. So we have some plant viruses. We did a little bit of SARS-CoV-2 work, but I won't talk about that today. So the content of the talk, I will start with a little bit of introduction on arboviruses and an older project to show how we study virus transmission. And that's a study about West Nile virus. Then we go into sort of these distinct environments that I'm talking about. So what is different about a vertebrate host cell and what is different about a mosquito host cell? And how does that affect the RNA composition of our viruses? And how does that affect the replication and of these viruses in these different hosts? And now we've done this with a couple of viruses. We've used mostly single-stranded RNA viruses, and Zika here in bold is the one that I'll be showing you most of the data about. But we're now getting similar results with West Nile virus. We then turn to a totally different arbovirus, which is blue tongue. It's a double-stranded RNA virus, and I'll show you, hopefully I have some time to show you some results about that as well. Right, so first, what are arboviruses? So arbo stands for arthropod-borne, and these are any of these invertebrate blood-feeding arthropods that transfer viruses from vertebrate animals or humans to the next. Now, these are not just, these arthropods are not just picking up the virus and dropping it somewhere else. The virus actually enters their gut, then needs to infect or cross the barrier, the first cells of the midgut. And that's often a barrier for these viruses. So if it's a poor combination, so that, let's say the mosquito vector is not suitable to transmit this virus, this is often the barrier where it goes wrong. Then the virus infects the hemocell of the mosquito, so that's the circulatory system, and finally it ends up in the last sort of organ of the mosquito, which is the salivary gland, and that usually takes about 4 days to a week until it gets there, and only then is the mosquito capable of, when it takes another meal, it salivates a little bit and then it's capable of infecting the next host. Now, there are actually over 1,000 different mosquito species that are able that drink blood and are able to transmit viruses. And what makes a good combination is still very much unknown. So we know that Culex pipiens is, in Europe at least, our common house mosquito, and that's very good at transmitting West Nile virus. While some of the Aedes mosquitoes, mostly the more urban and invasive mosquitoes, like the yellow fever mosquito, Aedes aegypti, or the tiger mosquito, Aedes albopictus, those are really good at transmitting Zika, dengue, and chikungunya virus. Now, we also have other animals that do this. So we have Culicoides species, which are these little midges, little biting flies, and they, for instance, transmit blue tongue virus. And of course, we have ticks, and tick-borne encephalitis is one of the main viruses that they transmit. So let's focus a little more on the flaviviruses, and the 2 viruses that I use mostly in the lab. So we use West Nile virus and Zika virus. And if you look at the the tree here, you can see that there are slight differences, and that's the reason why we chose these two viruses as model viruses. So on the top, we have the Japanese encephalitis cluster, which includes West Nile virus. And these viruses infect birds and are transmitted by Culex mosquitoes. But both these birds and these mosquitoes really inhabit urban areas. And these mosquitoes really like to drink from humans as well. So in that way, the virus ends up in humans. And if you're unlucky, it can cause encephalitis and you can die. While the Aedes mosquitoes for Zika, dengue, and below there in the tree, there's another cluster with yellow fever. They are much more urban and they like to feed more on humans. And you see that these viruses have a direct cycle between primates and humans and these type of mosquitoes. Now, in between, this genus has some clusters with insect-specific viruses. And one of them, we'll get to talk about that very briefly later, sits in between these mosquito-borne viruses and is still very closely related to these. So that's an interesting model group as well. Now, for West Nile and Zika, these two viruses, I think, are well known. We already talked about West Nile. Zika, we know, especially in pregnant women, can have horrible outcomes for the baby. So babies are born with underdeveloped skulls, which is called microcephaly. Now, both these viruses in the last sort of 20, 25 years have been spreading from Africa out into the rest of the world. So in 1999, West Nile virus was first found in New York and quickly spread across the Americas. And in 2013, we had the Zika outbreak starting in Brazil. And before that, Zika was just some insignificant virus. And the only thing we really knew about it was that there was one crazy virologist who wrote a paper saying that it was an arbovirus, but it was also sexually transmittable because he went home to the US and gave it to his wife, which is a really fun read, by the way. So these viruses, they have a single-stranded RNA genome of positive sense. And in that way, they're actually quite simple. There's one big polyprotein that's translated directly from them, and that's then cleaved into a capsid, an envelope protein, and a number of non-structural proteins. So as an example of how we study sort of virus transmission by mosquitoes, I want to take you back to 2010. So this is when in Europe, all of a sudden, there was an outbreak of West Nile virus in Greece. And this turned out to be a lineage 2 West Nile virus, which before then was thought to be only mildly pathogenic. But here in Greece, we saw actually that lots of people developed encephalitis and quite a number died. So from there, we also realized quickly that it was transmitted by Culex pipiens, the common house mosquito. Now, on the map here, you see these red dots. So that's where the outbreaks occurred. And we really wanted to ask the question, if our main mosquito in Northern Europe, in the Netherlands, is also Culex pipiens, is there a risk for it to come to us, or are there genetic differences between the mosquitoes, or is there something else? So very simply put, our entomologist neighbors, they made a really nice colony of Culex pipiens just straight from their backyard. And we infected these mosquitoes and mosquitoes from other locations with different isolates of the virus. Now, the way we do this is we have these little buckets. They have a gas top. In these buckets, we have the mosquitoes. And in this way, we transport them into our BSL-3 facility. Then you see on the left, on the bottom, there is a little metal cup, and it has a parafilm over it. This parafilm is used as fake skin, really. And in that cup, we fill the cup with blood and virus mixtures. And the mosquitoes mistake this parafilm as a skin. And if you put it on top of this little bucket, they will bite through it and they will drink the blood. Now, the only way they do this is if you heat it up. So that's called a hemotech feeder. And on the right, you see that's where we screw in this little metal cup. And that heats it up to a nice 37 degrees. So that the mosquitoes actually think they're drinking from a real human. Then you give them an incubation period. So we usually use a standard of 2 weeks. And then you knock them out. Because what we really want to know is there virus in their saliva as well? So we want to know if there's virus in the mosquito, but we also really want to know if it's specifically in the saliva. And so the best way to do that without having any other sort of tissues of the mosquito present is to keep them alive and harvest some of their saliva by giving them a drink. So the only way to do that is we knock them out with CO2, and mosquitoes are amazing creatures. They can lay on this pad that is oozing CO2, so displacing the oxygen for hours, and they have no negative effects. And what we then do is we pick them up and we take off all their legs and their wings so they can't go anywhere. And then we stick their proboscis into this little yellow tip that has a sugary liquid. So from there, they'll be just lying there, they have a drink, and they salivate in this, and from there we can isolate the virus. So that's the best sort of proxy we have for looking at the possibility of virus transmission. Now, if we did that with different mosquitoes from different areas and with different viral isolates, there wasn't any difference between these mosquitoes. But what we did notice, and then you turn your eyes to the map again, the background of Europe is colored, and that means the average temperature of the two warmest months of the year and peak mosquito season, July and August for us. And you see the Netherlands is really green, and that means it's sort of well below 20 degrees in this map. And if you look at the graph, this is how many mosquitoes actually got virus in their saliva. We see that the infection rate at 18 degrees is really poor. It's only 20%. But if you move it up to 22 degrees, 23 degrees, you get to 40, 45%. Now, if you look closely at the map, on top of it, it says 2011. For the whole of this study, which was published in 2015, this was sort of the case. But shortly after, climate change became visible. And you see now that the Netherlands is turning yellow. And that means actually we're hitting 20 degrees regularly and over that as well. So the last sort of 5 years, we've been hitting 22 degrees. Now we have a decade worth of this data. And if we look at the red data points where the outbreaks are, what the temperature there was, it's always above 20 degrees. So that 20 degrees seems to be the limit for West Nile virus to be efficiently transmitted by these mosquitoes. And that's not strange because obviously mosquitoes aren't warm-blooded, so they're sort of ambient temperature, and at higher temperatures, these things just work more efficiently. And the quicker a virus goes through a mosquito, the more chance it has to transmit it before it's eaten or dies. So it only took a year later that a consortium that we're a part of for the first time actually found West Nile virus in the Netherlands. And this sort of illustrates how important it is to do these type of studies and to keep looking for these type of viruses. Because before this, we would never screen in the Netherlands for West Nile virus because it wasn't there and it wasn't coming there. And for instance, Australia now had, like last year, Japanese encephalitis, a new genotype. So this always activates arbovirologists to look into this stuff, and this is really the reason why. So we had about a dozen human cases, and they were all identified retrospectively from cerebrospinal fluid. So these were all encephalitic cases. So we have no idea how many other cases there were really, but usually that's about 100-fold. Right, so then on to what we do now mostly. And that's, we really want to know, how do these arboviruses manage to replicate both in this human and in this mosquito? Because if you look at their last common ancestor, these two creatures are separated by at least 700 million years. And that's visible in a lot of ways. So one of them is in humans, we see a very acute infection. So we get sick for a week, maybe 2. And then if we don't die from encephalitis, we clear the infection. While mosquitoes, once they're infected, it's persistent, and the rest of their life, they'll fly around and they can infect other animals. Now, one of the main differences that we have, obviously, is our immune responses. So innate immune responses, we really heavily rely on interferon response, and then also that activating and strengthening a cellular response. While mosquitoes, they do have remnants, something similar to this interferon response. They have a JAK-STAT pathway, IMD and TOL, but they don't seem to be incredibly functional when it comes to virus infections. They mostly rely on RNA interference. But in both cases, what is absolutely crucial is how do these cells recognize the viral RNA to kickstart this innate immunity. And the most sort of popular theory behind it is, the question is, how do these viruses really balance between these two? Because you can imagine if a virus acquires a mutation that is a benefit in one, it's very likely not a benefit in the other organism. So what we first did was sort of compare the RNA of the hosts first. And we can look at just the 4 mononucleotides. We can look at codons, but what has the most striking differences is actually dinucleotides. And dinucleotides is nothing more than just 2 nucleotides in a linear molecule. So on the right, you see this yellow sort of single-stranded RNA molecule. And from left to right, there is a UPC dinucleotide, and in bold a CPG dinucleotide, and then a GPU, etc. So obviously we have 4 nucleotides, so we have 16 potential dinucleotides. And I put that P in between because that relates to the phosphate backbone of the molecule. And that's just so if you talk about a CG, you might be confused with a CG base pair, and we're talking about a linear molecule. So that's why I'll always say CPG. And if you do that, if you analyze this, and in the graph here, I've analyzed this for 10,000 randomly selected human mRNAs. And we look at the observed amount of dinucleotides versus what we expect. And what we expect is simply just from each mRNA, we count the amount of mononucleotides. And if you imagine putting them in a random order, you expect an X amount of dinucleotides. So that is set to 1. And what is immediately striking is the 2 black bars. CPGs are incredibly suppressed in human mRNA. They're only 40% of what you would expect. And also UPAs are suppressed to about 60%. If we do the same for a mosquito, so this is Aegypti, we see a very similar pattern, especially the UPAs are suppressed. The only thing that really stands out is these CPGs are not suppressed at all. There is some logic behind this. So in these organisms, obviously the RNA is made from DNA, and CPGs in DNA are easily methylated. And the methylated C can deaminate and turn into a T. So there is mutational pressure there, and these CPGs disappear. DNA methylation doesn't actually occur that much in a lot of these insects. So on the right, we'll see different groups of insects and their DNA methylation level. And if you look on the left in that graph, you see the Diptera, the flies. That's what the mosquitoes also belong to. And they actually don't show any DNA methylation activity at all. So that might explain why in their RNA, there's no bias towards CPGs. So that's all fun. What about these RNA viruses then? These are RNA viruses, single-strand RNA molecule. There's no DNA stage. There is no knowledge of the CPGs being methylated or otherwise mutated. But if we analyze them, and these are the mosquito-borne ones, we see they closely mirror the composition of human mRNA. So the question really was, why is that CPG suppressed in these RNA viruses? And this is something we see across the board from single-stranded positive sense RNA viruses. So also vertebrate-specific viruses. Now, in 2017, a group first identified a factor that might cause this, and that's the zinc finger antiviral protein. This protein has 4 zinc fingers, and it actually are shown to bind specifically to a CG dinucleotide or CPG dinucleotide. Now, the theory behind it is, as you see here, so if you have a wild-type virus that is well adapted and has reduced the CPGs, there's no binding and nothing happens. If you, for instance, would make a mutant of that virus by introducing CPGs, or you have a virus that is just poorly adapted to this host, this ZAP protein will bind. And while ZAP has no enzymatic activity itself, it can stimulate RNA degradation by sequestering some cofactors. It can block RNA translation with other cofactors, and it can stimulate interferon response as well. And it can even just by sitting on the RNA, we've seen that it will block virus replication. So perhaps just interfering with the RDRP, we're not quite sure what happens there. So for multiple vertebrate viruses, this has been done. What we do is then you just take the virus and you synthesize a piece of their genome where you synonymously increase the CPGs or the UPAs. And so with synonymously, we mean we leave the amino acid sequence of the proteins completely intact, so that this very simple figure at the bottom, you have a wild-type virus, the genome looks different, but the outside of the virus is 100% identical. And what we always see, or in most cases, what people see is if you do that, the virus becomes attenuated. And if you stick it in cells with where the ZAP protein is knocked out, for instance, at least the CpG high viruses are rescued again and they replicate properly again. So that really shows that this ZAP protein attenuates these viruses by binding these high CpG regions. But now what about these viruses that continuously switch between these two different hosts? So this graph actually shows sort of the same data as we've seen in the histograms earlier, but now the gray cloud are all the individual mRNAs of the human, and the black cloud are all the individual mRNAs of the Aedes aegypti mosquito. And we see on the x-axis are the CpGs, and we see that the human cloud is still severely suppressed, of course, and while the cloud of the Aedes aegypti is unbiased. And both of them severely suppress UPAs. Now, I've put it like this because then we can, on top of this, we can plot all the different flaviviruses. So in green, we see the mosquito-borne flaviviruses, and Zika there is indicated. And like I showed before, they all look quite similar to human mRNA. Now, what is interesting, I find, is that there's two clusters of insect-specific flaviviruses that are still closely related. And those are the orange and the red. And specifically the orange, those are the ones that in the phylogenetic tree that we saw earlier sit between the mosquito-borne viruses. So they are really closely related to these. And the most popular and most plausible theory is that these used to be arboviruses, but they just somewhere forgot how to infect a vertebrate host. And they don't need it because they are now just transmitted within the mosquito population. The question is, so we see that they are drifting to the right. So we see that they are gathering more CpGs. Is that just genetic drift because there is no bias, or is there some form of pressure pushing them to the right? Now, I'm not an evolutionary biologist enough to do this computationally, so we came up with some experiments to do this. So we made mutants of Zika virus and also West Nile, either increasing UPA, so up, or CpG, so to the right. And the way we did this, here we have the Zika virus genome. It's actually only in an area under the blue block there, which is about 16% of the whole genome. In that block, you see how many CpGs and UPAs are there naturally. So that's 53 and 57. And we add either 59 CpGs to make that area unbiased. And that one we call CpG 1.0. So that's the ratio of 1, unbiased. Or we added 190 CpGs, and that's the maximum we could do without changing amino acid sequence. And we did the same for UPA, where we went for the maximum immediately. Now, as a control, and I won't be talking a lot about these controls, but we always take them along, we scramble the sequence as much as we can without changing the amino acid sequence, and that will tell us if there is any sort of RNA structure that we don't know about or any RNA sequence that is important for virus replication. So it just validates for us that we can mess around synonymously in this region. Right, so first we stuck these into vertebrate cells, into human cells. And the first thing that was striking was that the CpG high viruses didn't do much. So in the left graph, these are A549 cells. The red one is the one with high CpG. And you see it's slightly attenuated, but only very slightly. And there's also the CpG, the unbiased one, the 1.0, and that actually replicates the same as wild type. Now, for other vertebrate-specific viruses, usually with this type of CpG enhancement, you get about 2 to 3 logs attenuation. So that's quite severe. And here's only 2 to 3 fold. So somehow these flaviviruses are a bit more resistant to this, but that's fine. And for UPA, we see a nice attenuation, which we expected. We do see in the ZAP knockout cells that all of this has gone away, so that's great. But we were looking for slightly more sensitive assays, and that's here we see that with the plaque size of these viruses. So we see the wild type and the scrambled in blue, plaque sizes are similar, but then if we start increasing CpG, the plaque sizes get smaller, and UPA is even smaller still. And in the ZAP knockouts, the plaque sizes were smaller to begin with, but they were similar across the board. So we then thought, okay, let's see what happens if we stick this in a vertebrate animal. And this assay, this experiment was sort of had two ideas. So first we wanted to know what would happen, how they would behave, these viruses. But secondly, also, can we use this as a vaccine strategy? So we first infected or vaccinated mice with all these different mutant viruses. And what we saw in the titers is that actually, similar to the cell culture, both the CpG max and the UPA max viruses had about a 7-fold reduction. It wasn't great, but it was less than wild type. And also in symptoms, so ruffled fur and weight loss of these mice, we saw a lot. A little bit, like they were a little bit healthier, but we still saw some symptoms there. Now, this was a sublethal model, so all these mice recovered. And then they were challenged with a pathogenic Zika virus. They were all vaccinated well, so once you get Zika once, apparently these mice don't get it again. And only the ones that were vaccinated with PBS, they showed Zika. Now, what is really nice about this model is it's a male mouse model, and we know Zika crosses like into the reproductive tract. And in this male mouse model, actually, it also crosses there and it goes into the testicles. And the testicles here become severely inflamed when they're infected. But after this has resolved, they actually shrink. So the testicles are less than half the size of a healthy mouse testicle. So we see the ones that were vaccinated with PBS and then challenged with Zika, or the ones that were vaccinated with a wild type or scrambled, have very small testicle sizes, while the ones that were vaccinated with our CpG max or UPA max viruses actually still had healthy testicle sizes. So that does show us that even though we saw a good amount of virus replication, there is some potential here to protect them from like severe clinical symptoms. But so the thing we were most interested in was what happens if we stick this in a mosquito. And I put all my money actually on the fact that, well, it's unbiased, so nothing, right? It will replicate the same doesn't matter if we increase CpG or not, it will do the same thing. So actually we saw the opposite. We see here that there's 5 different mosquito cell lines at a single time point, because I think this is a slightly better graph than a growth curve. But we see that the wild type and the scrambled viruses, they show very similar virus titers. But both the CpG 1.0 and the CpG max, or the high CpG viruses, showed a significant increase in virus replication in these mosquito cells. And the UPA max did do what we expected, that was still a little bit attenuated. So that actually shows that there is some sort of a drive towards more CpGs, or at least more CpGs replicate better. These viruses replicate better in mosquito cells. So of course we took this into live mosquitoes in our assay. So we infected them with Zika, with the Zika variants. Then 14 days later, we collected saliva, and then we checked first the bodies, are they positive or not? And then the saliva, are they positive or not? Now these are the mosquito bodies. So for Zika, it's quite normal that not all mosquitoes become positive. And we see that about 25% of the mosquitoes became positive. But in the CpG high ones, this was massively increased to above 60 and 70%. And if we look at the saliva, the difference is even larger. So it's 12% were wild type, but it's above 50% for CpG high versions. So as sort of a one-side summary, that really shows that if we mutate the CpGs in these viruses, we can actually attenuate it a bit in vertebrate animals, but we can have enhanced replication in these mosquitoes. And that sort of shows that these arboviruses do have to deal with opposing pressures from these two organisms. Now that we know this, we are really interested, and we have a number of other questions. So here, one of the questions that we had was, what happens with other arboviruses? And we went all out on a limb to go to the sort of the strangest virus in this respect we could find, which is double-stranded RNA. And I'll tell you why. And that's because, to my knowledge, nobody has done these sort of assays with a double-stranded RNA viruses. And that's because they don't seem to suppress CpG dinucleotides. So this is a paper that came out last week. And on the left, you see positive sense RNA viruses. And this is what I've been talking about more the whole time now. And in this paper, they actually didn't look at arbovirus, but they looked at invertebrate-specific and vertebrate-specific viruses. And you see in the green-blue, the vertebrate ones have suppressed CpGs, while the invertebrate ones don't, or far less. And this always correlates with the GC content. And we're not quite sure why that is, but what it means is just the higher the GC content in an RNA molecule, the more CpGs it can tolerate. When we look at double-stranded RNA viruses, we see that whether they're invertebrate or vertebrate RNA viruses, there is no suppression of CpGs. But most of these are segmented viruses. And if you look closely, there are quite a few dots that do show suppressed CpG. So a few dots that are underneath that cloud. And logically, like these viruses, double-stranded RNA is, of course, an enormous danger signal to cells, right? Because cells don't make it. It's always viral. So they hide their double-stranded RNA genome quite well. But they still have to make mRNA, so they still have to translate protein. And they still have to use the cellular ribosome and the machinery to do this. So if those come out with high CpG, wouldn't that activate the same system? So to test this hypothesis, we went to blue tongue virus. Blue tongue virus is transmitted by these little biting flies, these midges, between ruminants. It's an arbovirus. It has 10 segments of double-stranded RNA. Now, this is Ran, and she has done all of these experiments, so they're very fresh. She's still working on this a lot. But what she first did was overexpress ZAP in cells and see what happens. So on the left, she took a virus that we know is very sensitive. This is alpha virus Symbis virus. And you see that if she overexpresses ZAP, this virus becomes a bit attenuated. And there's two isoforms of ZAP that she used, ZAP-L large and ZAP-S small, and both function the same way. Another virus, which is our negative control, VSV, is insensitive to ZAP. We know this also, and she sees the same thing. Now, she did it with the blue tongue virus, and actually, she saw that it is a little bit sensitive to overexpression of ZAP. So from there, she went on to make a knockout cell line. So she knocked ZAP out, infected the cell line with blue tongue virus, and that's the red bar. And you see there's at least a 4-fold increase in virus replication. When she then reconstituted ZAP by transfecting in plasmids, and keep in mind, she's not hitting all the cells. So there's a lot of cells that still are ZAP knockouts that don't have ZAP, but she does get a good sort of recovery of that antiviral effect. And on the right, those 4 bars, those are actually overexpressed ZAPs as well, but they have a point mutation in one of the zinc fingers that are known to stop them from binding these CpGs. So they're actually our best controls, and we see that those have no effect whatsoever. So really, this CpG binding of ZAP, this function, is important to inhibit arbovirus replication. So it's not just blue tongue virus IF20, but she's done it with a couple of blue tongue viruses and also equine hemorrhagic disease virus, so all arboviruses, and she sees that they all benefit from having ZAP knocked out. Now, so how do these viruses, if that's the case for all of their 10 segments, that is highly inefficient. So we hypothesize that they must have some way of dealing with this ZAP antiviral response. So if we look at all the different BTVs, there's 26 or 27 here, and the 10 segments that they have, there's one segment in blue that shows mild suppression of CpGs. And this segment actually codes for non-structural protein 1, and that protein is known to be needed very early on in infection to stimulate translation of all the other segments and replication of the virus. So the logical hypothesis is this protein is expressed a bit better than the other ones because it has less CpGs and RNA, and maybe interferes with ZAP. So what she did, she used that Symbis virus that is very sensitive to ZAP, and she transfected in her knockout cells one of the ZAPs and this NS1 protein. And what you see then is that if you don't add NS1, there is quite a bit of inhibition of Symbis virus. If you add NS1, there's a lot of rescue. So Symbis virus does a lot better in the presence of blue tongue virus NS1. So that really suggests that this is a molecule that antagonizes ZAP. So this is still ongoing, and she's still working on this, but I think it's a very interesting piece of work. And then finally, one of the questions that we are really interested in is not just the ZAP molecule and the CpGs, but actually are these UPAs. Because all organisms, they seem to suppress these UPAs. And the best theory that we have is that UPAs destabilize RNA. So suppressing them gets you a more stable RNA, but the exact cellular factors that are involved in this are still a bit fuzzy. So there's a few theories. So you have A and U rich regions that are usually attacked by a number of proteins. There's also endonuclease RNase L, which cleaves RNA after a UPA, but also after a UPU. And UPAs are suppressed, but UPUs are not. So there are some holes in all of these theories. So we're really interested to see what goes on here. So for that, we have developed an RNA binding assay. So this RNA affinity purification assay is simply put, we couple bait RNA, single-stranded, to a streptovidin bead. We run a cell lysate over it. It can bind to whatever the bait RNA is. And then we use it from mass spec to identify what is binding here. And because this is quite artificial, it's outside of the context of any sort of virus or coding sequence, we can really muck around with how many UPAs, how many CpGs, how we alter these RNAs. And it's showing to be a really nice way of looking for binding proteins. So if we do this and we focus on these UPAs, what binds more to UPA high proteins generally are all components that have something to do with the spliceosome. Now, this is also still a work in progress, so we're still digging through this. And this is still a large cloud. But like I said, we can do multiple different RNAs with the same composition. We can do it with infected cells, with non-infected cells. And in that way, we can reduce our cloud a little bit. And on the left, you see a slightly smaller cloud where these are the ones that are still present in all of the different conditions that we tested. So we're still digging through this massive heaps of data here, what the best way is to analyze it, to look at it. But still, the spliceosomal complex, that's the one that seems to be very much involved. Now, if you flip it the other way around, what does not bind to UPA high RNA might be equally important. And here, actually, I think it's quite striking, is the ribosomal proteins that do not bind. So what we get is these ribosomal, so translation initiation might be less for RNAs with high UPA. And also there's a lot of proteins that have a function in sending these newly deformed proteins to the ER for further post-translational modifications. And that is, I think, the data that I really want to end on. So of course, we're using this as well to figure out what is happening with these CpGs in the mosquito. But for this, we had to adapt the system a little bit, so we're not quite there yet. But that's definitely ongoing. So the conclusions is that we see very distinct CpG usage between vertebrate hosts and invertebrate mosquitoes or arthropods. The Z protein detects these high CpGs, and it's even functional against double-stranded RNA viruses, where NS1 or blue tongue might be a good antagonist. For UPA, these components of the spliceosome might be involved, and/or translocation to the AR, ER, and RNA translation. Now in the mosquito, of course, we get better replication with high CpG. We can play around with this. For applications, we can think about making mosquito-specific viruses more pathogenic, for instance. That's something we're working on to wipe out mosquito colonies with a natural-born virus. But another thing that I like to play with the idea, but it's highly unethical, I know that, so is to use mosquitoes as flying doctors. So we could actually make a CpG high virus that goes very well in a mosquito, release them, and let them vaccinate bird populations for West Nile or whatever. Of course, this is a silly idea, but I still like it anyway. And maybe for some sort of animal virus in the future, it might have an application, who knows? Yeah, so I really want to thank these people, the people from my lab, Sander Koenraad, our neighboring entomologist, also Andreas Suurbier from Brisbane, who has been a great help with the mouse experiments that I showed here. And with that, I'll take any questions.
Thanks, Jelke. I'll just start off first. So if I understood that correctly, we would want evolutionary pressure or pressure on the virus to basically be higher in CpG so that they then get recognized and destroyed in the host cell, as in vertebrates, and then remain in the mosquito.
Yeah, as an application, you mean? Yeah.
Yes. And so something similar to ZAP or something like that. Could you introduce that into the mosquitoes?
if you could introduce it. Probably.
Or like, yeah,
Yeah.
breed them. I know there's like in malaria, they're trying to crossbreed mosquitoes and things like that.
Yeah, so you can now make transgenic mosquitoes, and you probably could. But I think there's something we kind of want to do these experiments just for the sake of it, is to see if you have an attenuated virus, right? The levels in the blood are lower, and these mosquitoes need a certain level to actually pick up the virus. So an attenuated virus as a vaccine would probably not end up in a mosquito, even if you're bitten the next day, except when it replicates perhaps much more efficiently. So these are experiments that we like to do still. But I would think probably we don't have to worry. I don't think it will end up, if we ever use a live attenuated vaccine like this, I don't think it ever ends up in a mosquito.
Okay. So I thought that was really interesting. We had a lot of research here on dengue, so hopefully that was useful for a lot of people. So just put our hands together and thank Jelke for a great seminar.
Thank you.